Seems silly, but it's actually really important. So on this type, if you look at this one, see how it's got this black kind of a thicker edge? All that's showing you is that where you see that thick edge, that's just saying we're looking at it from this edge. So you can see our viewpoint is onto that plane, and it also doesn't mark the carbons. So if you look on here, there is no carbon on that point. But whenever you see this hexagon or a pentagon or whatever it happens to be based on the number of carbons, literally you just have to say, where's the first carbon? This one happens to have an oxygen here. So we're going to go to the right. There's carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. We can actually identify this by saying, oh, this has 2 hydroxides at C1 and C2. So that tells you what this looks like based on C1 right here and C2 right here. Okay, you may have gotten that in chemistry. If you did, that's a review, and if not, that's how you read this chemical formula. A disaccharide, obviously di is 2. So when you get 2 of the monosaccharides together, we call that a glycosidic linkage. So when we link them together, it says you the answer. I was going to try to hide it, but I can't. So this is dehydration, which means we pull a water out. When we pull a water out, it leaves these 2 edges having nothing there, and they have an energy deficit, and therefore they are going to want to, this oxygen says, I need another bond. So it needs another bond in there, and it will bond to the other carbon that is over here, because remember, each of these is a carbon, and all carbons want 4 bonds. So if we go back here, I just want to show you, this will work. So if we go back here, each of these carbons has 4 bonds. This first carbon has 1, 2, 3, 4 with that double bond. This carbon has 1, 2, 3, and 4 bonds. And that is, if you did well or you remember chemistry, that is because carbon, all of these are the noble gases over on the right hand side. The noble gases are perfect, and the structure in which their electrons are in is also perfect. So these do not need to have any change in their electrons in order to be stable. Everything else on the periodic table needs to have some sort of a change or an alternative, on the amount of electrons they have in order to be like these noble gases and be perfect. So carbon's right here. Its ionic state would then be 1, 2, 3, 4. It's 4 short of being perfect like neon, so it could be okay, cats, you can't do that right now. So it would be a -4 because it's 4 short of being neon. Or in this case, it could also, you can't see the other half of my periodic table, sorry. You could go backwards and go 1, 2 beryllium, 3 lithium, and then 4 helium, and it could actually be up 4 electrons above helium. So if it loses 4 electrons or gains 4 electrons, either way, it becomes perfect. It always needs to have 4 bonds. And the fact that it can go above or below to the nearest noble gas makes it very, very flexible, and thus it becomes the the basis of all carbon living forms, because carbon is very, very flexible that way. Nitrogen would be 1, 2, 3, a -3. Oxygen is a -2, and it's too hard for oxygen to go back and gain and have 6 electrons to be helium, so it rarely does that. And fluorine, of course, is only one electron away from the same configuration as neon, and therefore fluorine has a high electronegativity, meaning it's going to be highly attracted to electrons because it only has one thing to change, one electron to gain, and it will then be perfectly stable. And that just personifying it kind of makes it so it tries really, really hard. Okay? All right, so now get that lined up. So now we are back to our ring forms. That's where you get these numbers. And remember, when you're looking at structures, here's oxygen. It's supposed to have, it's a -2. Should have 2 bonds. It does. One with carbon and one with hydrogen. Okay? This one has one with carbon, the one with the hydrogen. Same all the way down here. All right, moving on. Disaccharide. So here's what we're going to be adding them together. Here's 2 glucose. We take a hydrogen out. That leaves 2 ends where it's unstable energy. So it's either a deficit or it has too much. And therefore that oxygen that used to be attached to this glucose now also has the other bond. Remember, it needs 2 bonds. One is right here with this carbon, and the other one is with this carbon. This is perfect. Now we have this bonded together, and we made maltose by adding 2 glucose together. If you like malt balls or malt shakes, this is maltose. It's a different kind of sugar, even though it's just 2 of these put together. Okay? And another way to do this is by adding glucose and fructose. Fructose is really sweet, sweeter than glucose. And if we take a water and a hydroxide and a hydrogen and pull water out of these 2, then we can make them bind as well, and we end up with sucrose, which is table salt. So table salt, of course, is just gritty, actual just sugar, sugar, sugar. So it tastes really, really sweet, and so does fructose. Glucose is sweet, but not as sweet as fructose. Here's a little thing. It was in my chapter 10 book that I read. It talked about high fructose corn syrup. High fructose corn syrup was because they found a way to take the glucose in corn, and by changing it around, the arrangement, we can actually turn it into fructose, and therefore it tastes sweeter. You don't need as much of it because it's sweeter tasting, and corn is pretty cheap to grow. So now we have high fructose corn syrup, which frankly, just like old commercials used to say, which you probably don't remember, but yeah, it's high fructose corn syrup, but it's just got sugar in it. It's just sugar from corn. So it's still just a sugar. It's just there to sweeten things. It's when our foods have too much of it in it that we end up with some issues. Okay? Polysaccharides. We already did that. So now we're going to talk about some polysaccharides. So now we're going to talk about polysaccharides that are able to, like I just talked about the potato, actually store a bunch of sugars. And because of the way that they're arranged, we actually get some different kind of structural strengths, I guess, if you will, from the way that they look. So, starch. Starch is a large group, though, of glucose monomers. So we're going to end up with plants are what make these, and we're going to end up with polysaccharides. You guys know saccharide is a sugar, and a bunch of sugars in plants where they're going to be all glucose, and we are going to put them all together, and we're going to store them. And as we store them, they actually combine with each other, and they make starch. So this is starch in a plant. This would be like corn. Corn is a starch. Corn is a vegetable, yes, but not as good for you as something with less sugar in it. So starch, or potatoes, those are starch, or sweet potatoes, those are starch. So what they're doing is they've actually taken in polysaccharide, and when they take it in, it puts it into these, where's my other picture of it? Okay, it's right there, but hang on. So it's going to put it into these helixes. And this helix is a form where we can store, just like the cord on a telephone that's circular, it's wrapped up like a landline. Sorry, well, I'm aging myself here. But a landline that's twisted actually has a lot more surface. It's a lot longer than it looks because we twisted it and put it in a helical form. We can store a lot in there because we're using this different type of form. So this is, starch is an alpha form. And then this is another way, so that we're doing the same type of thing. We're going to actually take sugar in the form of glycogen. We're going to put this in your muscles. So in your muscle, we're going to leave this in the muscle itself. And if right now at this second, whatever you're doing, you had to run and run and run and run just to save your life, these little packages of starch, these little glycogen packages are sitting in your muscle, and they are really close to the lovely mitochondria. The mitochondria can grab those, they'll go into the muscle and give you energy right there. And it's a starch, so there's lots of sugars in it. You're going to have lots of energy. Adrenaline's going to help you grab those, and then give you lots of energy to be able to use your fight or flight mechanism to either run and get away or to stay and fight. For those of you who want to stay and fight instead of running, that's totally up to you. So this is starch. This is a form of starch. Plants do it. We do it by putting it together and keeping it in these little pockets inside your muscles. Generally, it's just kind of how it is. The more you use them up, then the body tends to store more of them. So if you're an athlete, you tend to, I'm sorry to use the A word in this, but if you are an athlete, sorry DSA, then you keep these, you keep more of these glycogen granules, more of these packets available. And as an athlete, because you've used them more, you will also have more mitochondria available because you use them all the time. Mitochondria are one of those things that if you don't use, they tend to go away up to a certain point. Everybody has mitochondria. Those who work them out tend to have more. Okay. Glycogen, we already got that. So I want my picture of that, but we'll do that in a minute. So the other version of a starch is cellulose, and cellulose is a little crazy. So for this, we have our two different forms. We have an alpha and a beta form. So this is the alpha form. I don't know why that's a question mark there, but that's okay. So this is the alpha form. The alpha shows the hydroxyl group right here on the bottom of the ring. Remember this darkened little section right here that looks 3D shows you that we're looking at it at that plane, and it's showing the hydroxide on the bottom. The beta form shows it at the top. You think this wouldn't make much of a difference by simply switching the hydrogen and the hydroxide around, but it does. So what it does is it's going to make it so that with this is going to be like starch. Starch has all hydroxides lined up on one side. Because of that, we also have the other side where they're not lined up. There isn't any on that side. So this tends to go in a helical form. Like I just showed you back here, you tend to have this form in a starch. It's spinning around like DNA type of thing, because this way we're storing more, well, I shouldn't say that. This way the hydroxides are now able to connect to each other because they're all on one side. We have to spin in order to be able to see the rest of the hydroxides and be able to connect with them. So in order to do that, we're going to have to use these. This is called 1,4 linkages. So this is number one carbon, right? There's an oxygen. That would be 1, 2, 3, 4, 5. This is a pentose. It's got 5 in there. So now carbon 1 and carbon 4 are connected together. Starch is a 1,4 linkage of alpha glucose monomers. The beta, so these are beta where we have the same thing. We're going to have a 1,4 linkage, but now On a couple of our glucoses, we actually have that hydroxide is at a different spot. So because it's a different spot, this is actually, if you notice, turned upside down. So this is like a mirrored image of it. It's turned upside down. And now we're going to be able to, notice there's one on every other side. We're going to be able to combine directly with the row of cellulose right next to it, which is going to make it very, very strong, almost like a 2x4, where we're going to have this row, a row right next to it. A 2x4 is basically a piece of wood, 10 trillion toothpicks all lined up in the same direction next to each other, and that's where you get your strength. So that's what we're doing with this. We're going to have them line up next to each other. And by doing that, so betas, there's our betas. By doing that, we get an unbelievable amount of strength, which is why this would be cellulose. You can't break down cellulose because it's in these rows, and these rows are very tightly knit. If we were trying to pack them in, if we put every other one upside down, then they're going to fit even closer. Like when you put shoes in a shoebox. You put one in with the heel or one side in one direction, put the other shoe in the other direction, and they're going to be able to fit more shoes in the same shoebox. That's what we're doing. So we are going to get this super tight knit. It's in these straight rows. It is so strong, you can't break it down. Cellulose is what you find in cell walls of plants. So these cell walls are so strong, and it's not just in leaves, it's in lettuce, it's in carrots, it's in apples, it's in oranges, it's in spinach, it's in zucchini, it's any vegetable or anything from a plant that we eat goes into your body. Your body's enzymes come in and go, oh, sure, we can break it. We can't break it down. Oh, we can't break it down. I feel like we just went in a weird direction with this, but let's just keep going. So we can't break it down. We can't break it down. We can't break it down. And they can't break cellulose down. And so what does your body do with anything that it cannot break down? It just sends it straight through the digestive tract. This cellulose is fiber. When they say you need a high fiber diet, they say you need a high cellulose diet. This is cellulose. It comes in these strands. It goes in these microfibrils, and you can't break it down. So we end up just having this go straight through the body. So think to yourself, why would this be good for a diet? You think, and I will have a drink of Sprite. Why would that be good for your diet? To have something go through your body and you really can't get hardly anything from it. We can't get anything from it. So sometimes when you chew it, if this was a spinach leaf, let's say, and you chew it, you might be able to break it down and get some of the iron from it and get some of the trace minerals from it, but you're not going to get any carbohydrates from it. You just can't. These are your carbohydrates, right? Monomers linked together, and they're so tightly linked, we just can't break it down. This is going to help everything else go through your body. It's like a little street sweeper that goes through the digestive tract and clears everything out. And that is really, really important. They say the more fiber you eat, the less chances you have of getting colon cancer. And colon cancer's percentages in the U.S. are climbing drastically because our diet has changed so much in the last 60 years that colon cancer is now much more common than it used to be. This needs to be added. So what about animals that only eat cellulose? How are they getting anything from it? A big cow can only eat cellulose. Well, he can't go through and break that down. That's going to be really, really hard for him. Oh, there we go. Herbivores, cows to termites. So the cow has to have a bacteria. Actually, a cow has a protist. No, I think it's a bacteria that is in its stomachs. Yes, it has 4 different chambers. So it chews it, spits it back up in its mouth, chews it again, chews it again. They eat rocks. The rocks go in and help break it up. They break it up as much as they possibly can. And then at the very end, to break down that glucose, they have bacteria that will actually help break it down. It's a symbiotic relationship because without the cow bringing in this already kind of mushed up plant material, the bacteria would never, I still want to say it's a protista. It's a prokaryote, so it could be a bacteria. Then the bacteria would never get the food, and the cow would never be able to get any food from the grass, so they work for each other and helping each other. Sometimes cows even eat what they poop out because there's still so much energy left in there that they could eat it again and again and still keep getting energy from it. Termites. Termites do not eat wood because wood is made of, that's right, good job. Wood is made of cellulose, and cellulose is this, what we're talking about. This is cellulose. Termites can't break this down either, but termites can keep, I think they have a protist that is inside their gut, and the protist, they bring them the wood. The wood gets broken down by the protist, which gives them sugars to the termite and gives sugars to the protist that is actually breaking the food down for them. So they all get to have this symbiotic relationship that helps them break down this cellulose. Okay, so the last one is chitin. So chitin is another polysaccharide. Chitin is found on the outside of all bugs. It's found on the outside of crabs, lobsters, anything that's going to shed its skin.